Good evening, students, and welcome to Tute's live stream. My name is Sarah. Welcome to our live GCSE revision session, dedicated to helping you excel in your Biology Paper 1 exam tomorrow. This evening, we're diving into effective study techniques and key insights designed to boost your confidence as you prepare for tomorrow's exam. Joining me this evening is
Hi, I'm Kate. I'm one of the other science teachers here at TUTE. I'll be answering any of your last minute questions. So please submit these questions through the link that I'll provide in the YouTube chat, where I'll also be offering some exam specific tips directly into that chat box. Okay, thanks, Sarah.
Thank you, Kate. Cheers. Okay, so welcome to the lesson, everybody. So you can see from our opening slide, like all our Tute lessons, it begins with a learning objective. And the learning objective today is what do you need to do to prepare for your Biology Paper 1? So let's get started. Okay, so as usual, there are 3 learning outcomes. The first is to explain how best to tackle different questions on Biology Paper 1. And in addition to that, we're going to practice a relaxation technique to support you during your exams, because we know just how stressful a period this is. And it's good to have some little strategies up your sleeve just to put yourself back in the zone and recollect yourself in the event in an exam situation. And then we're going to describe briefly the structure of the Biology Paper 1, but just what you see when you look in. So that you know what you're going to face tomorrow and it doesn't come as a big surprise. You know what you're going to be up against. Okay, so let's take a look at our first question, which is on respiration. So the question is a typical question that you might see at the start of an exam. Which of the following is the word equation for aerobic respiration? You can see it's got 1 mark and you've got 4 options. So this is a classic opening question you might see tomorrow. Now, what I put there is a bug symbol, and underneath it, the word bug with 3 words next to it. The first one is box, the next one is underline, and the third one is glance. And you might be wondering what on earth that's all about. But this is actually a really good strategy that you can use in your exam to support you tomorrow. And what it basically means is box means identify what the command word is in any question you're asked. So in this case, the question is which, but it could be why or where or how or explain or calculate, whatever that command question is. And it's good to be focused on exactly what that is. So you can box that, and you can do this with a pencil on your paper. And then you need to underline key words in this question to make sure you're absolutely crystal clear about what this question's about. Because one of the mistakes a lot of students make in exams is they don't read the question and they end up answering the wrong question, which would be a real shame because you'll miss out on those marks. So by underlining these keywords, it makes sure that you're crystal clear about what is being asked about and you really thoroughly understand what that question is asking you. And then glance is simply to glance at the mark scheme to make sure that you're alert to how many marks are available for this question. Because if there are a lot of marks for it, then you want to put more in it. But if there's only a couple of marks or 1 mark, you don't want to waste a lot of time. You don't want to write reams of stuff for questions that are 1 mark. You just want to put a short, succinct answer. So that mark scheme guides what you are, the amount that you're going to write, and stops you writing too much for questions that only are worth a couple of marks. So this is a classic multiple choice question. A top tip with all multiple choice questions is to read all the options in the multiple choice carefully, because it's very easy to suddenly see an answer, think, oh, I know it, that's that one, without reading all of them and choosing it without realizing there might be a better answer further down the list. So basically what you really want to do is read every single one and make sure that you know at the end, right, I know which one I'm going to choose. And of course, the one you want for this one, the word equation for respiration, is this one, which of course is glucose plus oxygen goes to carbon dioxide and water. Okay, so glucose, of course, is what is the fuel for life. That is what drives respiration. Oxygen is the gas that it reacts with. And we are getting glucose from our diet. We get it in our food. We get oxygen from the air we breathe in. And that oxygen and glucose is carried around our body, in our blood, to all our cells that need it. And in our cells, the mitochondria are where the little engines in our cells that carry out this reaction, that allows us to generate that energy and produces carbon dioxide and water as a byproduct. So that is that word equation, and it's really important that you know it. Okay, so the second part of this question is asking, it's setting the scene, it's setting up a sort of scenario, and a lot of questions in your exams do this. So it says some students were investigating the effect of exercise on an athlete's body, and they collected data on their heart rate before and after the exercise. So what you're presented with here is a table, and tables often come up in exams, and the expectation is that you will be able to interpret what's in that table. So it's important to pay attention to what the table is telling you. So on the left-hand side here, we can see that what we're being shown is heart rate, which has been measured in beats per minute, volume of blood, which is pumped out by the heart in each heartbeat. So every time your heart beats, the exact volume that your blood of blood that's going to be pumped by your heart, and then something called the cardiac output in centimeters cubed per minute. And right there, you've got a clue about where that comes from, because the heart rate is beats per minute, and the volume of blood that's pumped by the heart each time is measured in centimeters cubed. So really, this is the rate of how your heart pumps out blood in 1 minute. It's centimeters cubed per minute. It's important to be alert to units. And you can see here we've got two sets of data. The first one is resting heart rate and resting volumes of blood being pumped out, and the resting cardiac output. So these three figures represent what is being measured for someone at rest. And you can see that these during exercise, these numbers change, and unsurprisingly, they go up. So we have heart rate during exercise has gone up to 125. The volume of blood pumped by the heart each time has gone up to from 90 to 145. And the expectation is that the cardiac output would do the same. So we've had a good look at that table, and now we're going to analyze the results. So the questions now are, by how much did the athlete's heart rate increase during exercise? And there's 1 mark available for this. So this is a straightforward calculation that you need to do. And again, you need to go to your table and you need to identify the variables that you're interested in, which are the heart rate when you're resting compared to the heart rate during exercise. So this is a simple calculation that you need to do, where you need to subtract the resting heartbeat from the heartbeat during exercise. And if you do that, you're going to get 125 minus 65, which gives you 60 beats per minute. Okay, so that is the difference. That's by how much the athlete's heart rate has increased during that exercise. The next question is an explain question, where you're asked to explain why the athlete's heart rate has increased. So again, you are thinking bug, you're thinking, box the word that is the command word, say why the athlete's heart rate increased. That's what you really want to identify here. So obviously, when you are doing exercise, your heart rate does need to increase. Why is that? Well, basically the answers that you need to give, and it doesn't have to be all of this, but these are suggested answers that could secure those 3 marks. So any 3 from to carry more oxygen and glucose to the cells quicker, or so more respiration can occur to release more energy. So you're saying why and what effect that'll have, or to allow more muscle contractions, because obviously if you're doing exercise, your muscles are contracting. The more you do, the more contractions your muscles going to have to do. They need energy for each and every one of those contractions. And then the other thing, of course, your blood has the job to do is remove the waste products of respiration. That carbon dioxide is actually a poison in our body. Your blood takes that carbon dioxide away and brings it back to our lungs, where we breathe it out. Okay, so of course having a faster heart rate will facilitate that, to make sure that carbon dioxide is removed from your body effectively. So top tip in a question like that, be careful to explain your answers as if asked why, so that you're absolutely giving the examiner what they want to see and securing those 3 marks. Okay, so the next question is a calculation question. It's asking, calculate the cardiac output in centimeters cubed per minute for the athlete during exercise and show you're working for 2 marks. Okay, so they're being quite explicit here. They're asking you to show their marking, and it's always show your working. So it's always a good idea to show your working, because that helps the examiner see that you know how you went about getting the answer. And even if you didn't get the right answer, if you put the wrong answer in numbers into your calculator, they would know that you had actually tried the right method, and you will still get one of those marks. Okay, so what we have to do here is we have to look at the heart rate in beats per minute during exercise. We look at the volume of blood pumped out by the heart in each of those beats. And basically what we know is that the cardiac output is the centimeters cubed pumped out per minute. So you know how many beats and how much each of those beats is going to produce in volume. All you have to do is multiply the two together. And if you look at the resting data, you could probably figure out from that how to calculate this cardiac output, because it will be the same calculation. So all you do is do your sum, 125 multiplied by 145, which gives you 18,125 centimeters cubed per minute. So you're off and running. Then just you've got your figures, you're going to get your marks, because you have 2 marks going for this question. 1 you'll get for showing you're working out, and that's why that's so important. And the second is if you get the correct answer, which in this case is 18,125. And just be alert to your units. The units are given to you in the table, so if you just need to replicate those in your answer. So yeah, just take care with those calculations and show you're working. Okay, so the next question is, give 2 other changes that occur in the body during exercise. And again, this is quite straightforward. The mark scheme is telling you it's 2 marks. You're being asked for these changes that occur during exercise. Well, think about any time you've had to run or catch a bus or whatever it is where you've had to carry out physical exercise. It always it has other effects other than your heart beating faster. And those other things that can happen, of course, are your breathing rate will increase, or the depth of your breathing will increase. You will breathe deeper, and then there'll be an increase in your body temperature. You might even start sweating to lower your body temperature. And another thing your blood vessels can do to facilitate you during exercise is that the blood vessels going to your heart can dilate. That means they become wider. And because they become wider, they basically allow more blood to get to all those cells in your muscles that need that supply of oxygen and glucose to keep them going and keep you running as fast as you can, etc. So that 2 of those suggestions. You don't have to do all of them, and that's something to remember. Don't waste time writing lots of suggestions. You only need to make 2 to secure those 2 marks. So yeah, top tip, be careful to explain your answers if asked why. Okay, so basically, we're going to look at a slightly different scenario here, where as the exercise becomes more vigorous, the athlete's cells switch to anaerobic respiration. Okay, so we're asking the question here, why would human cells respire anaerobically? So why would the cells switch to anaerobic respiration? Well, this girl here is doing a sprint hurdle. She's going to use a lot of energy very fast. It's going to be a struggle for her, even with her heart beating faster and breathing deeper, to get that oxygen and glucose to her cells quick enough. And it might be that they can't quite do it. And basically, in that for a short time, and it's only a short time, your body cells can switch to what we call anaerobic respiration. Well, how does that work? Well, basically, it works. Why would we do this? Because, and there's 2 possible answers here. It might be because not enough oxygen is present for aerobic respiration. And like I said, the body has a hard time delivering that blood with all enough oxygen and glucose, especially if you're doing super strenuous exercise. Or you could say that more energy is required for exercise than is available from aerobic respiration alone. So the aerobic respiration isn't doing enough. But the body can resort to anaerobic respiration so that they can keep going for a short time. Okay, so yeah, top tip again, be careful to explain your answers fully. Okay, so The next question is name the chemical produced in anaerobic respiration that is not produced in aerobic respiration. So we know from our first question, the multiple choice, that basically when we have aerobic respiration, glucose reacts with oxygen to produce carbon dioxide and water. But there is no oxygen in this reaction, so we just have plain old glucose as our reactant. So what is produced instead? Well, you need to be aware of this key term. This is what is produced in the absence of oxygen. It is lactic acid. And that's all you have to say. You just need to know that lactic acid is produced. And for that, you will get your 1 mark. You don't need to write more than that, because remember, 1 mark is all there is available for this question. Okay, so the next question is to explain how the substance produced, because they're not telling you, you have to figure that out, is broken down after the exercise is finished. And basically, this is a 2-mark question. I should also point out, this is a question that you would be asked at a higher level. You would not be asked this question at a foundation level because there isn't an expectation that you would know this. So if you're doing a higher exam, this is something you need to be alert to. So lactic acid, which is produced during exercise, does need to be broken down after exercise. And that's one of the reasons we have what we call the oxygen debt, because basically the lactic acid is carried back to the liver. And that's what you need to say for 1 mark. So that lactic acid that builds up in your muscles, it has to be removed and taken to the liver. And that, during this time, you might experience some cramp. That's what one of the symptoms are of buildup of lactic acid. So the blood taking that away helps relieve that cramp. And basically then, when it gets to the liver, then oxygen, and that's where your oxygen from your oxygen debt is required. You basically keep breathing hard after your exercise so that oxygen can be brought to your liver to oxidize the lactic acid, and that it converts it to glucose again. Because one of the things we know about anaerobic respiration is it doesn't produce as much energy as aerobic respiration. Why? Because the glucose isn't fully broken down in anaerobic respiration. It just gets as far as lactic acid. But you can build that lactic acid back up to glucose in the liver. Okay, so guys, top tips for that question: read all the options, multiple choice carefully, read table data carefully, be clear on what it's telling you, take care with calculations and show you're working, because that can get you marks even if you get the question wrong, and be careful to explain your answers if asked why. You need to give a reason, say what's happening and why. Okay, guys, so we're going to move on to another question on digestion now. So food molecules such as proteins cannot be absorbed unless they are digested. Why is that? Okay, so this is just a 1 mark question. Right, well, what have we got here? We've got a great bit of steak here on the barbecue. Your body cannot possibly assimilate that like it is as it exists. So that's the job of your digestion system. Your digestive system, from the moment you put something in your mouth, you're chomping it up into little bits, you're mixing it with saliva, you're lubricating that food so it can pass easily down through your esophagus, down to your stomach, and all the time it's going to be pushed and pummeled by the peristaltic contractions all the way along your digestive system. And basically and then of course you get the rush of enzymes to break it down. And that's because we've got to break this great big bit of meat down from that huge protein structure into individual tiny molecules. And that's because those tiny molecules are essential, because the big protein molecules are just too big to be absorbed by our body through the walls of our intestine, and they're insoluble. And basically one of the things we are doing with digestion is converting these big insoluble big molecules into little individual tiny molecules which are soluble, and when they're soluble they can be absorbed across the wall of your small intestines. Okay, so the next part of this question is asking, proteins are digested by protease enzymes. So what does protease break proteins down to? So again, this is just a 1 mark question. You just need to say one thing to secure that mark. You don't need to write masses. And we know that proteins are big structures, but the little building blocks that form a protein are called amino acids. So amino acids are the building blocks of protein. When you digest a protein, a big meaty protein like this you'll have in this food, you will break that down from these big protein molecules into individual amino acids. And those amino acids are soluble and can be absorbed readily across the wall of your small intestine. Okay. The next question is, where is protease produced in the digestive system? Well, protease, because this is a serious digestive job, is produced in 3 parts of your digestive system. And those 3 areas that you need to be aware of are the cells in the walls of the stomach. So the protein digestion begins in your stomach. Okay, it's no other food group is broken down in the stomach, just the proteins. Okay, so that tells you proteins, they take a lot of breaking down. It needs to start right early on in the stomach. Okay, and then that when the food leaves the stomach, it goes into the small intestine. But of course, next to the small intestine, we have the pancreas. And although the pancreas isn't a part of the digestive system, it has this really critical role where it releases all these digestive enzymes into the small intestine right at the start, which can include protease enzymes. And that immediately starts the work of digestion or continues the work of digestion on those proteins. And then the food passes on through the small intestine, and cells in the walls of the small intestine are also producing proteases. And your small intestine is really long, coiled up in your tummy, so that you can have enough time to break down all that food. The digestion of these foods takes time, and that's why your small intestine is so long, because it allows time for those digestive enzymes to be produced and break that food down. Okay, so the next question is a bit different. It's saying, how do you test a food for protein? Okay, so this is one of the required practicals. You need to be aware of the different types of food tests for proteins, carbohydrates, and fats. And the food, and you also need to be aware of the different food tests for starch and sugar. So if you're not sure of those, that's something you might want to check out this evening, because those food tests quite often come up. But the one we're interested in here is protein. Okay, and this is a 4-mark question. So you have to say, clearly and simply what you would do if you were testing a food for protein. And what you do is you grind up the food into a small beaker and add distilled water. And this is what you do with all foods that you're testing. By the way, you start with this first stage. You could take that food, you grind it up in and with pestle and mortar or something, and you add distilled water. What does that distilled water do? Well, distilled water means that when you stir, some of the food dissolves into that water. And then what you can do is you can filter that dissolved food to get rid of all the debris, all the chunky stuff, okay? And then you're left with a solution which will contain some of that main food group that the food groups that you're interested in. So those first 2 stages are the first stage with all the food tests that you would carry out, whether you were testing for carbohydrates or fats, and those carbohydrates could be sugars or starch. So you then take some of this solution and put it into a test tube, and you add a solution called Biuret solution, which is basically a combination of copper sulfate and sodium hydroxide. But you don't need to worry about that. Just say it's Biuret solution, and it's blue. It's a pale blue solution. Okay, and if there are any proteins present in that solution that you filtered, then that Biuret solution will change color. It will go from blue to pink or purple, depending on how much protein there is there. So this little set of test tubes illustrates the kind of results you would get if there was no protein present in the food. It would not change color. It would remain pale blue. But if any protein is present, it will change to either pink or purple, and then you know you've got protein present. So it is a top tip. Be clear on the outcome of the food tests, like the Biuret test. You need to know the iodine test, the Benedict's test for sugars, and maybe the Sudan test or the ethanol test for fats. So make sure you're aware of those tests. Okay, so the next part of this question is the pH can affect the activity of different proteases in the breakdown of protein. In an experiment, the effect of pH on the digestion of protein was measured. Okay, so this question is linked to this topic. It's asking what is the optimum pH for the protease enzymes A and B. So what we've identified here is two enzymes that are responsible for the digestion of protein. And the data here reveals the pH at which these two enzymes work best. So with any graph, it's important to read your axis. You read a graph just like you read anything. The way you read a graph is you read the x-axis, and you can see that the x-axis is showing pH, and it's showing it increasing up through the pH scale. And then on the y-axis, you've got the rate of enzyme action. That's how quickly the enzyme is acting, okay? And you can measure that in an experiment, okay? And that, again, is one of the required practicals, guys, looking at the effect of pH on the rate of digestion. Okay, so we can see here we have two different curves for each of these two enzymes. So we can see right away that they operate differently at different pHs. So what they're asking here is what is the optimum pH for the protease enzymes A and B? And to figure that out, well, basically you need to see where are they working best. And the y-axis will tell you that because where they peak is where they are working best. And basically you can see here that A is peaking here, and all you do is drop a line down to the x-axis to find out the pH at which that enzyme A is working best. And then you repeat it for B. You move across and see where it's behaving best or operating best, and you draw again a line down to the axis, and it will give you the values at which that enzyme is working best at whatever pH that is. And from this data, we can tell that the optimum pH for protease enzyme A is 2. And the optimum pH for protease enzyme B is about 7.5. Okay, and you can read that data off your graph. And if you give that those correct marks, just take care when reading your graphs and drawing graphs, because it's actually quite easy marks to get. You don't need to know anything. You just need to be able to read that graph accurately. Okay, so where is this next question is asking where? So there's our box word. In the digestive system is the protease enzyme produced, and this is for 2 marks. Okay, so there's the protease enzyme A is clearly working best at an acidic pH. And there's only one place in your body where you have that very low pH, and that is your stomach. Okay. And so the stomach, the protease enzyme is produced in the stomach where the pH is 2. And that's because, and this is what you need to say for your second mark, that the stomach contains hydrochloric acid. And that's where this protease enzyme A operates best. So that's the 2 marks for that question. You just need to say those 2 things. Protease enzyme is produced in the stomach because you need to be aware of the fact that the stomach is very acidic, and that's where protein digestion begins, and that's because it contains this really strong acid, hydrochloric acid. Okay. All right, the next question says, why does the rate of enzyme action decline if the pH is higher or lower than the optimum pH? Okay, so basically, you can see here that as soon as the pH gets less or more for both enzymes, the rate of enzyme action drops off considerably. And that's because enzymes are very sensitive to changes in pH. If they're put in more acid conditions, that acid will react with that protein and it will change its structure. And equally, if the pH is too high, like more alkaline conditions, that will react with that protein and cause it to change. And of course, that is a bad news for an enzyme because basically it'll change the structure of that enzyme. And what we know about enzymes, of course, is that if that happens, the enzyme is damaged. And the word we use for that is denatured. So that's an important key word to use when we're talking about enzymes being damaged by too high a pH or too low a pH or too high a temperature. It denatures. And what we mean by that is that the protein or whatever the food element that it is digesting can no longer bind to that enzyme's active site. And that's because, as we know, enzymes have this very particular shape, which allows a substrate molecule like a protein or a carbohydrate or whatever's been digested to latch on exactly. So if that enzyme is damaged because of too acidic or alkaline conditions, it knocks out that active site and those food molecules can't attach anymore. So the reaction cannot be catalyzed and it means the reaction might stop. So that's a serious business. Our enzymes are critical to everything that we do, including digestion. And if something damages our enzymes, it makes all those processes impossible to carry out. And that's why these optimum pHs are what our body works hard to maintain so that our digestive system can work effectively. Okay, so yeah, when think about experiments are trying to show, be clear on what each of these required practicals are demonstrating, and that'll help you understand and explain the results. Okay, so top tips for that question: read the question carefully so you are clear what's been asked. Be clear on the outcome of food tests like the Bayerette test. Take care when reading or drawing graphs. They're actually easy marks to get, but you have to take good care so that you make sure you read those graphs right or draw them right, etc. And think about the what experiments are trying to show. So it's easy to read a methodology and go, oh, you do that, but why? Why are you doing that? What's it showing you? What's it telling you? You need to be clear on what it's actually trying to demonstrate. What is the experiment trying to show? Okay. And on that point about experiments, it is important that you pay attention to the required practicals. If I was giving you advice tonight, the night before an exam, it would be to go over those required practicals. And Kate in the chat box, I know, is sharing with you the details of the 1, 2, 3, 4, 5 required practicals that might pop up in paper 1 tomorrow. And there's little links there to YouTube videos that you could watch, which are only 5 or 6 minutes at most, that will tell you about them, what they're about, what they show, the very things that you might be asked about tomorrow. So if you've got half an hour, you could watch all those videos. That would be a super preparation for your exam tomorrow. Okay, so at this point, we're going to take a little breather. We're going to do a breathing exercise, which will help be something that you can use as a little tool now, tomorrow morning, during the exam, after the exam. So these are breathing exercises. Now, basically, when your body is stressed, you kind of breathe more shallowly, and your body begins to think, is something up? Or is our supply of oxygen depleted? We know already from the respiration question how important oxygen is. Well, guess what, guys? You've got to reassure your body that everything's A-okay by breathing. And then it goes, all right, we're okay. We've got enough oxygen. That's actually the most important thing. We can still breathe. Never mind the exam. We've got our oxygen supply. Everything's good. So with this breathing exercise, you basically start here. What you do is you breathe in for 4 seconds. Then you hold it for 4 seconds. Then you breathe out for 4 seconds. And then you hold it there for 4 seconds before you start again. And by doing that exercise, and it's easy to remember because it's just 4 things: breathe in, hold, breathe out, hold. You will be amazed how that helps recenter you and gets you ready for the next question. You could do this between each question, and it puts you in a much better place going into that next question. It doesn't take very long, like 16 seconds, but it could make you calmer and more prepared to answer more questions. Okay, and the third question that you might be asked would be about osmosis. Osmosis often comes up on practicals questions, and of course, it's one of the required practicals. And here we're being asked to describe. So here's our boxed off word, the command word, describe. They want you to describe a method you would use to investigate the effect of different concentrations of a salt solution on the mass of pieces of potato. And this is a major question. It's got 6 marks, but don't be put off by that. You just have to say 6 things, guys. That's all it's asking you in a nice, clear, concise way. They don't have to be big, super long sentences. They just have to be clear and concise. So osmosis, of course, is all about the movement of water in and out of cells, okay? And you need to be clear on what osmosis is. It's the movement of water from an area of high concentration of water to an area of low concentration of water. And it always happens across a semi-permeable membrane, okay? And basically, tissues, the maintenance of the water concentration in your cells is very important to how they function. And if they're put in solutions where they are, which are very strong, like salty solutions, then your cells will lose water and might shrivel up, and that's not good. But equally, if they're put in a too dilute solution, then lots of water will go into your cell, and they'll fill up and maybe even burst. So that's not too good. So what you want in your cells is just the right thing, where the water is moving in and out of your cells equally, because the water's always going to be on the move. It's just going to be moving in and out equally, so your cells are nice and stable. So osmosis is an important process for all our cells to maintain that balance. So if you're carrying out this experiment to look at the effects of different concentrations of salt solution, which could radically alter the movement of water in and out of your cells, then this is the sort of equipment you could use: a weigh balance, a ruler, test tubes for all your different concentrations of salt solution, a knife to cut up your potato samples. Okay, so like I say, write short, clear sentences when describing methods. And here's a sample of what you could write. Remember, somebody should be able to read these instructions and follow them if they were doing the experiment themselves. So you would say the investigation I would do would be, firstly, cut 5 pieces of potato into equal size pieces. Okay. Then weigh each piece of potato. Then put each piece of potato into different concentrations of salt solution. So they'll be varying in concentration from very dilute to pure water, right up to much stronger solutions of salt. And then you leave the potato pieces for 24 hours to let that osmosis process take place. Okay. And then you remove each piece of potato, dry it, just tap it, because you don't want to soak up the water or affect the results. You're just getting the residual water off. And then you reweigh it. And then, having reweighed it, you can calculate the change in mass of each piece of potato. Because depending on whether they were put in a really strong solution where they might shrivel up, or they're put in a very dilute solution where they might expand, that's going to affect the size of that potato and the mass that it'll be after the experiment. Okay, so what are the control variables in this investigation? Well, we know the variable that we are changing is the concentration of the salt solution. The thing that we're measuring is the change in mass. But every other thing in any experiment, you should only either change and measure one thing. Everything else has to be kept the same. And that's what we call the control variables. All the other things you need to keep the same. So what are the control variables in this experiment? Well, basically, we want to be controlling the length of time that you leave those potatoes in the solution. Now, we've said in our methodology that it's 24 hours, and it should be 24 hours for every one of those pieces of potato, right? It shouldn't be more or less for each one of them, because that could affect the results. If they're left in slightly longer, one's taken out a bit earlier, that could affect your results. So that time has to be exactly the same. Then the shape and size of the potato you cut has to be exactly as close as possible the same, because you don't want the shape and size that you cut to affect how water moves in and out of that piece of potato tissue. And then temperature can affect all sorts of experiments. So we want to make sure that those potato pieces are all kept at the same temperature. So those are examples of control variables that you want to keep the same, but it's basically every other single thing that you're doing apart from the thing that you're changing, in this case, the concentration of salt solution, and the thing that you're measuring, which is the change in mass. So yeah, short, clear sentences when describing methods. Okay, so basically this is the sort of data that you'd get from an experiment like this, and you could quite often be given this data in the exam. So you can see in this table, we've got the concentration of the salt solution in moles per decimeter cube. That's standard measurement of concentration. We have the mass at the start, and then we have the final mass. And then we can calculate the change in mass from the start to the final mass. So basically, this is a set of data that you could be presented with in an exam. And you just have got to take a good look at that data and figure out what it's telling you. And again, you don't have to study to figure that out. You can figure that out just by looking at it. You can look at the numbers and go, well, what are these numbers telling me? So the first question here is to calculate the value of x. And you can see here, x has been identified in the table. And basically, x is the change in mass. So they've done it for all the other variables. So you actually could copy, you just have to say, well, what did they do in this one? Or what did they do on the one below? And I need to do the same thing. So that's common sense. So basically what you need to do here, of course, is you have to look at the final mass and the starting mass. And then what you have to do is subtract the two. So the final mass is 2.98 and the initial mass was 2.71. So we know that this piece of potato that was in a zero point 0.2 solution gained 0.27 grams when it was put in that 0.2 solution. So basically water was going into that potato at that concentration. Okay, so the concentration was of that solution was obviously more than the concentration inside the potato, so that fluid was going in. Okay, so basically, and just remember units, of course, but the units are always spelt out for you in the tables as well. So two of the numbers for the change in mass have a negative value. There they are. Okay, what do these negative values mean? Now, the question here is only 1 mark. So you have to write something very, very simple to get these marks. You don't have to go into the why they're negative values. You just have to say, what does the negative value mean? Well, the negative value means simply that instead of increasing in mass like our 0.27 piece of potato did, they are decreasing in mass. So they are losing mass. And that's all you have to say. The potato lost mass. And bingo, you've got your 1 mark answer. Okay. All right. So very often with a set of data, then you might be asked to plot a graph, and you use the data from the table to draw. And the other thing you're being asked here is to draw a line of best fit. Now, again, you don't need to know anything to get these marks. You just have to take the data from that table, plot it, and draw a line of best fit. You don't have to have revised anything. You just need to do that, plot that data. So these are 3 marks that you could get with just by drawing a graph accurately. So take care when drawing points and graphs and line of best fits, because these are marks that you can readily get. Okay, so you just take the data and you plot it. Now we have the table, and you can see what I hear is here's the first point, and here's the second point, and here's the third point, fourth and fifth point, and then having marked those points and use the scale, remember, on the x-axis and the y-axis to make sure those points are bang on correct. So read your scales really carefully to make sure you're putting the points in the right place. Then having got your set of points, you draw a line of best fit. Now it's a line of best fits, which means it's a straight line. Sometimes you're asked for a curve of best fit, and then you draw a curve, but it'll specify which one they want. So basically, a line of best fit is a straight line through those points. And you want to make sure that that line of best fit goes through as many points as possible. But if it doesn't go through all the points, because sometimes it can't, just try to make sure that you've got as many points above as below. So for example, this one's a little bit above, this one's a little bit below, but provided most of the points are, the line goes through most of them, you will get that mark. So just take care, make sure you've got a pencil and a ruler, of course, guys. Bring a pencil and a ruler as well as your pens into the exam, so you can do that with pencil and paper. Don't be messing around with biros, because you might need to rub something out, and you need to be able to change your answer if you're doing a graph. Pen for the rest of it, but for graphs, use a pencil. So which solution of salt solution would give no change of mass? So this is an interesting question, because the point at which there was the concentration of salt which would give no change in mass is not one of the measurements that you actually have in the table, because where you get no change in mass is where the points are going through 0, and this is it here. So basically, you have to identify from the graph what the concentration would be that would result in no change of mass. Okay, so that's what this is showing here. And that value is 0.52. Okay, so the next question is explain why there would be no change in mass of a piece of potato at this salt solution. Well, for 3 marks, what you need to say is basically it's the same concentration as inside the potato cells, and therefore there's no net movement, and that's an important word. Water is going in and out of these cells, but if it's at concentration which is the same as the potato cells, it's going to go equally in and equally out because the concentration is the same. And that's what we mean by net movement. So there's no net movement of water. It's going equally in both directions. So the potato isn't getting bigger, it isn't getting smaller, it's going to stay the same size. Okay, and that is by the process of osmosis. So those are the three things you need to say to secure those marks. Okay, so top tips. Write short, clear sentences when describing methods. Take care with drawing points on graphs and lines of best fit. Take care with analysing graphs. Always have a go, even if you're not sure. You might be right. That's an important tip. Always have a go. Okay, and don't spend too long on questions you're not sure about. So we've looked at tackling different questions. We've looked at relaxation techniques. Now, very briefly, I'm going to look at the structure of the biology paper. So this is what you'll be confronted with tomorrow morning when you sit down at your exam. And the information on this page of paper looks a bit dense, but all it's telling you is the materials that you're allowed to bring in include a ruler and a calculator. So if you haven't got your calculator yet, get one on the way in, because you can bring a calculator into all your science exams and your calculator maths papers. It says answer all the questions in the spaces provided. Yeah, don't do right outside. And do all rough work in this book. Cross through any work you do not want to be marked. Then it gives you the breakdown of the marks, of how many there are, etc., and urges you to use good English as far as possible. And in calculations, clearly show how you work out your answer. Okay. And then, of course, you need to put that important information onto your exam sheet, which your school will obviously have provided you with, and you want to make sure those details are nice and clear. So that's what you can do while you're waiting for the exam to begin. So the assessment of the exams is basically, you'll find that questions start easier and get harder as you go along, but it's a bit up and down. So I always call it the low-hanging fruit. Every question will have easy marks at the start. So you want to make sure you get through the whole paper to nab those easy marks, okay? And that way you'll max out the amount of marks you get. Okay, so, and obviously foundation papers go up to 5 and higher tier papers go up to 9. Sorry. Okay, so that's the structure of the paper. Okay, so we're now at the stage where we can take some questions. I'm going to, Kate's going to share some questions with me. So let's have a look. Yes. Okay, so here we go. Questions from pupils. What is the best way to tackle 6-mark questions? Okay, so what is the best way to tackle 6-markers? Well, 6 markers are very often things like methodology. So the key thing is you want to write 6 things, but you want to write 6 coherent things that really, you know, um, make it clear the examiner knows what you're talking about, and say them in a nice logical fashion with nice English. You don't have to write massive sentences, but you do want to try and make sure that you are basically saying everything the examiner wants to hear, and it can be nice, short, brief, and to the sentence, to the point questions or points that you're making. Okay, next question. Let me see. What is oxygen debt? Okay, so oxygen debt. Let me pop that in the chat here, or in the on the screen, to make sure everybody's aware of what question we're answering here. So basically, oxygen debt. Well, oxygen debt, when you're running a race or something, basically you are going to be breathing really hard, trying to get all that oxygen into your lungs and then into your blood. And then your blood is going to carry that glucose and oxygen around your body to the cells that need it, like your respiring muscle cells. But if, like we described with that question on respiration, not enough oxygen is getting there, okay, and your body has to switch to anaerobic respiration, that's because your body didn't have enough oxygen to deal with that lactic acid. So after you finish exercising, and sometimes for quite a long time after you have your exercise, you have what's called the recovery time, where you will continue to breathe quite hard because you're still trying to get that oxygen into your body that's going to go into your lungs and into your blood and travels to your liver, where it's going to allow that lactic acid to be broken down or to be converted into glucose. So of course that creates an oxygen. That oxygen debt is basically what you owe your body in terms of oxygen. It provides the oxygen that you didn't have enough of when you were doing your exercise. I hope that makes sense. Okay, so the next question is what is the difference between arteries, veins, and capillaries? Okay, so you have this amazing circulatory system, guys, an absolutely amazing circulatory system. We take so many things in our bodies for granted, but basically, so you and the heart, funnily enough, of your circulatory system is the heart, and basically there are vessels that leave the heart that take blood to around the body, and then as that and takes the blood to your lungs to pick up oxygen. So your arteries are the process, the business of taking blood around your body and providing everything, all your cells, what they need. And basically, those arteries have really thick, strong muscular walls because they're operating under high pressure and they have to be able to expand to allow that pressure of blood coming through. And then it gets that blood to all your cells, each and every one of your cells. And then, of course, when it goes in amongst the tissues, it splits and divides into tinier and tinier vessels until you get down to the tiniest vessels, which are the blood capillaries. So the capillaries are literally little vessels which have walls which are 1 cell thick. It doesn't get smaller than that, okay? And that's to allow the diffusion of substances like oxygen and glucose across the wall of those capillaries into your cells, okay? And basically so they have to be super tiny so they can get into each and every one of your cells, and your cells are bathed in tissue fluid where all these substances are being exchanged, okay? And it's not just substances being provided to your tissues like oxygen, glucose. Your cells, like in your muscle cells, are producing substances like carbon dioxide that have to be taken away from those tissues because their buildup will do your cells harm. So carbon dioxide will diffuse into those capillaries, and then it'll be carried away. And then as what happens to those capillaries is they start joining together into bigger vessels. And those bigger vessels are veins. So your veins are what carries that blood back to your heart, okay? And basically it goes back into your heart. All those veins get bigger and bigger, and you have the big old vena cava, which is the biggest vessel, and veins have slightly thinner walls than the arteries. They still have muscular, but they are wider. But the other thing they have is valves. Those valves stop the blood going the wrong way because the blood in your veins isn't under as much pressure. It relies on your muscles, etc., to keep that blood moving. But it also has these valves which stop the blood going backwards, make sure the blood goes in the right direction back to your heart. And of course, then the veins go into your heart, and then and of course it goes then to your heart, to your lungs again for reoxygenation, and picks up all the other food items, etc. it needs to as well in the digestive system as well. So arteries, thick muscular walls that can allow that expansion. If blood in pressure increases, and then capillaries are the tiny little vessels in your tissues which are thin walled and allow diffusion. Veins carry all the blood back again to your heart, and they have slightly thinner muscular walls, but they also contain these valves that prevent the blood going the wrong direction. Okay, so now I'm going to go to another question. What are the reactants of anaerobic respiration? Okay, so basically, anaerobic reactants of. So the thing about anaerobic respiration is it's all about the absence of oxygen. Okay, so I'm just going to copy this question so everybody's clear on what this question is. So here we go. In fact, I'm going to go to the next slide, give myself a bit of space, because to understand the difference between oops, wrong little thing. So text box. So what are the reactants in aerobic respiration? What I'm going to share with you here is actually, because I thought, some people might be a bit unsure about aerobic and anaerobic respiration. So I thought I'll write something down here that just makes it a little bit clearer for people who are not sure about that. So I'm going to make another little text box. Here we go. And I'm going to pop this answer in here, which I think might help answer this person's question. So basically, what are the reactants with anaerobic respiration? Well, the reactants in anaerobic respiration are just glucose because there's no oxygen. So where we had aerobic respiration, it's glucose plus oxygen goes to carbon dioxide and water. Okay. With anaerobic respiration, you don't have the oxygen. You just have the glucose. So the reactant is glucose. There's just glucose. There's no oxygen. And basically, what you get, the products of that anaerobic respiration, depend on whether you're an animal or a plant or a microorganism. So, anaerobic respiration takes place in the absence of oxygen. And anaerobic respiration in animals like us produces lactic acid, as we discovered in that example where we were looking at when that athlete had to switch to anaerobic respiration because they just couldn't get the oxygen to their cells quick enough. But in plants and microorganisms like yeast, the products of anaerobic respiration are different. So what is produced in plants is ethanol and carbon dioxide. And we actually use this to our advantage. Yeast is used for a process called fermentation, okay, which is how we make things like alcohol, like beer and wine. And carbon dioxide, of course, yeast is used in bread because it produces carbon dioxide that allows the bread to rise. Okay, so I hope that answers your question about anaerobic respiration. The reactant is just plain old glucose because there's no oxygen, but the products depend on whether you're an animal or a plant or microorganism. Okay, so just be aware of those different products. That's the thing that's really different in anaerobic respiration. Okay, so just going back to the questions, we're going to see how can you tell whether your line of best fit should be a curve or a straight line? Well, it usually says so. Like in that question, it'll say, and it's the other thing, even if it didn't say, it's usually pretty clear from the data that you've got like a curve. So basically, what you will find is that you are, let me just move slides. Pop this question in here so people can see it. Okay, there we go. Control V. Oh, no, that's the wrong one. We just get that question copied. So yeah, so basically it is just the fact that the data will speak for itself. When you plot it on the graph, you'll see pretty quickly what kind of data you've got because it will literally look like a curve or it'll look like a straight line. So that will be your best indicator for what is going on there. So don't be worrying about it too much. Your data will be clear enough. They'll give you reasonable points that will show it's in a straight line, or it'll have, a point which, points that clearly show a gradation and then flattening out. For example, if it was the photosynthesis reaction and the effect of light, very often you tend to find, if I can just draw a little graph of that, you'll find it would be points that would sort of go up and then curve over like that. And those points the data that you might be given will show that curve happening because and it'll show that the last couple of points are like literally in a straight line like that. So you go, yeah, okay, I get it. This is a curve because points go up and then they curve over and go flat. So and again, just take care, making sure that as many points are on one side of the line as are on the other. If you can't get an exact line, don't be trying to draw a line between them. They want a curve. They don't want you drawing lines between these different points. Okay, guys, well, I'm afraid that's all we've got time for our questions. So just to finish off, I just want to say thank you all for joining us tonight. We hope you found this session valuable for your exam tomorrow. Before we sign off, don't forget to join our next revision session, which you can access via the timetable in chat. Your feedback will help us make even better revision sessions. So please complete the form, which is pinned in the chat. Don't forget to subscribe and click on that notification button so you don't miss out. And thanks once again for being part of our revision session. Remember, you've got this, guys. Wishing you all the best in your exams. Stay focused, stay motivated, and keep striving for success. See you next time.